About media and methods for culturing algae. Last class, isolation of algae, and combine this media and methods for culturing algae. In this chapters are component of media for culturing algae: carbon, hydrogen, minerals, trace element, vitamin, and growth factors. These also the carbon source like CO2. Bicarbonates are organic carbon needed for photosynthesis or building in biomass. And nitrogens, nitrates, nitrites, ammonia. It is essential for proteins, nucleic acids, and chlorophyll. And minerals for cell structure, enzymes. It will produce an ATP. And trace element and zinc and molybdenum it needed for in tiny amounts as enzymes and cofactors it will be used. FeMn zinc is also. And next for factors for algal growth. What are the factors? Light. It is the intensity of light it require various. And source natural sunlight or fluorescence and tubes it will be used. And next for pH, what are the ranges between 7 and 9? Optimum 8.22 and 8.7 for most microalgae. It will be used for optimum temperatures. And aeration and mixing, so it prevents the sedimentations and gives uniform exposures. It benefits, avoids thermal stratifications and improve gas exchange also. And methods, mechanical aeration using is the using for the wheels and jet pumps also. Aeration and agitations method. And temperatures optimal 20 to 24 degrees centigrade for optimal temperatures, and salinity for optimal 20 to 24 gram per liter. This is important mainly for marine algae. It will be cultured. Next for stock solution, macronutrients and trace elements and vitamins concentrated the stock solutions are prepared and then subsequently diluted to them. This is the stock solutions diluted to get the final media and concentrations. And next culturing media, culturing methods. So what are the culturing methods? Indoor and outdoor. So lab controlled environment for indoor. It will be used as artificial light and controlled temperature and good for research and pure cultures. Outdoor barns and uses sunlight cheaper used for large scale commercial productions, and based on system open or closed, open system open bonds and exposure to air, low cost but risk of contamination and evaporations. Closed system photobioreactors, tubes, bags controlled, no contaminations, higher yield but expensive. Based on axenic and non-axenic. Axenic is a pure culture, only one species of algae, no bacteria and fungi. Non-axenic mixed culture, algae plus other microbes used in wastewater treatment. This is also the Bowles basal medium, BBM. This is the important for the freshwater algae cultured medium. It will be used for freshwater algae. For example, pH is 6.6. The component for macronutrients, trace elements, and ADTA KOH solutions. It will be prepared for 1000 ml distilled waters also. And how it is used for make separated concentrated stock solutions for macronutrients and trace element and ADTA and Fe. Diluted them 1x for the final working medium. Adjust the pH for 6.6 and sterilize before inoculating the algae. And next one, Benex broth. This is the soil algae medium. Benex broth also the soil algae medium. The composition for 1000 ml distilled water, make it the pH in 7.0 to 7.5. The simple than BBM favor components lower concentration. The soil algae used to specifically for isolating and culturing algae for final samples. This is the important for the soil samples. Next for choose medium. So this is also the blue green algae medium for and pH ranges 8.5 to 9.0. This is the soil algae medium. What are the trace elements? Stock solutions is very important, is prepared. Very diluted medium compared to BBM. The concentrated here much lower. Good for fresh water for phytoplankton. pH for 8.5 to 9.0. And the comparison for the media: bold facial medium, Panax broth, and choose number 10. The medium is also is presented. And next, walnuts medium. This is the marine water algae medium, marine microalgae. It commonly used for the isocyrase and Heteroceras algae is presented. And mainly nutrient stock as per the later for final medium. Make it the trace metal solutions and first stock, and vitamin stock is added in separately. This is the batch culturing. Batch culture. What is it? Batch culture. This is the single inoculation of cells into cultured medium. So growing period of several days, and final harvesting at once at the end. This is the definition for batch culture. And advantages for operational simplicity and easy to set up and run, and rapid remedy of defects if contaminations it happens for one patch. And another advantage is low cost, and disadvantages low efficiency and predictable quality. This is the disadvantages also. And batch culture's growth curve lack exponential, stationary, and death phase. So it will be harvested usually late exponentially, early stationary phase, or best quality. Next slides on continuous culture. So continuous culture, this is the maintaining a culture in a steady state by the regulated addition of fresh culture medium. How it works from the diagram: media reservoir, fresh medium pumped into pump. And second one, chemostat vessels, the culture tank where algae grow. An inflow of media, fresh nutrients come in continuously. Outflow of media and cells, equal amount of old culture plus cells flow and the cell effluent tube. And collecting receptacle, this harvested algae is collected. The concept exponential growth phase for long time. The cell density, pH, nutrients all stay constant. So advantages: this is a high efficiency, consistent quality is second one. And disadvantages: cost and complexity and risk of contamination and technical skill needed. This is the disadvantages. So quick comparison for three culturing media: batch is at the start, at the end. The media preparation, addition of the start, harvested at the end. This is the continuous culturing. Continuous culture continues at the addition of the medium, harvested continuously. And this is the left diagram for turbidity state. How it works: controlled by cell density and turbidity. A photocell measures how loud turbidity the culture is. When cell density gets too high, valve opens, fresh sterile medium flow in, excess culture flow out. And components: so reservoir of sterile medium, culture vessels with stirrer, light source and photocell to sense turbidity, and hemostat right diagram. So the component inflow of fresh sterile medium with flow rate control, culture vessel with stirrer for mixing, air sparging for aerations, outlet for culture. This is the component zone. This is the turbidity state and hemostat. The features: control, turbidity state, cell density, turbidity, and hemostat, nutrient flow rate. And growth rate, maximum not controlled is a turbidity state, and hemostat controlled by dilution rate, and cell density kept constant for turbidity state. and hemostat can vary, and uses rhabdostate maintain exponential phase, and hemostat study nutrient effects. This is the both have high efficiency and consistent quality, but rhabdostate is more complex and hemostat is easier to control. And next slides on semi-continuous culturing. Semi-continuous culturing, this it is the middle method between batch and continuous. So main points: fresh medium ordered at intervals. You don't add media continuously, you add only a few after a few days. This is the semi-continuous is the main points. A partial periodic harvestings also it will be used. And first one, day 1 inoculated, add to grow, and day 4th harvested at 25% and add 25% fresh medium, and add, and you can harvested and grow for 25%. After that 7 days harvested 25%, you can add 25% fresh medium, it can and so on further following the methods. So next advantages: better efficiency than batch and simpler than continuous, widely used in aquaculture. The advantages. And disadvantages: quality can vary, not as consistent as continuous culture. It still needs regular labor, harvesting and adding media manually. So batch is at least a media additions. So batch culture is a media is a addition you can added. So batch and semi-continuous and continuous, these also the culturing method for very important. Next slides on culture type. What are the advantages and what are the disadvantages? So differentiate. And continuous culture is best for lab. What are the biotech and pharma industries where consistent quality and matters. And next, semi-continuous best for aquaculture firms and outdoor barns. This is the semi-continuous. And batch research labs, teaching and starting cultures as a batch cultures also. Thank you. So these also the culture methods also very important, and general microbiology unit 3rd.